Bismillahirrahmanirrahim. Assalamu alaikum students. I am Rabia Ubaid from Army Public School and College Boys Potowar Complex Islamabad, and today I am going to start the first chapter of biology first year book, Cell Structure and Function, and this is the part of Hssc-I online sport program 2020. The chapter number 1 of your book is Cell Structure and Function, and the topic we are going to study right now is cell theory, and the second one is techniques used in cell biology. So this chapter is all about the structure, the function of cell and the organelles of cell. You have already studied it in class 9th, but here you will discuss each and everything in a detail. Objectives. At the end of the lesson, you will be able to understand what is cell theory and what are the techniques that used in cell biology. Techniques used in cell biology means what are the techniques that we can apply to the cells to study the structure, to study the function, to study the components of the cells. Cell theory. First of all, you must know, and you have studied it, what is meant by cell. Cell is the basic unit of life. Why we called it unit? Because we know that cells joins to form tissue, tissues joins to form organs, organs join to form system, and when different systems combine, they form an individual. So the basic unit of life where all the life processes are occurring is a cell. Cell theory. Following are the points of cell theory. Number 1, cell is the basic unit of structure and function in living thing, because all the functions and all the functions that are carried out by different organelles present in the cells are performed by cells. Like cell has nucleus, cell has mitochondria, cell has Golgi bodies, endoplasmic reticulum. All organelles are present in cell, and each and every organelle is performing its function. And that's how one cell, other cells, and many cells perform their own function, and they combine to form a big organ system. Second, all organisms are made up of one or more cell. Why we have said that one or more cell? Because you have studied that like bacteria is made up of only one cell. There are many organisms that are unicellular. By unicellular we mean made up of only one cell. And more cell, there are many organisms that are multicellular. So there are some organisms like I will mention example of bacteria. Bacteria is made up of only one cell. And we human beings, we are made up of million and trillions of cells. Number 3, cell arise from division in pre-existing cells. Pre-existing cells will, like already a cell exists, when a division takes place in that cell, it will give rise to a new cell. It is like, I will mention example of bacteria. You have studied binary fission. The bacteria, the one cell of bacteria break down into two, and from that one cell, two daughter cells are formed. So new cell will arise only by the division in already present cell. Number 4, cell carry genetic material to pass to daughter cell during cell division, and that genetic material is DNA. When cell divides, first of all its DNA divides into two, and when the cytoplasm or cytokinesis take place, the new daughter cell formation takes place. That copy of genetic material transfers from parental cell to the daughter cell. So that is how DNA genetic material is transferred from parent to the daughter cell. Fifth point, all cells are essentially the same in chemical composition. Yes, all cells are the same. They have the same content of cytoplasm, and the cytoplasm contain the same content of water and other nutrient. 6, energy flow occur within cell. Energy flow, cells within the cells, the energy flow occurs. Now the next topic is techniques used in cell biology. What are the techniques? Basically, what is meant by this? Techniques that we use to study the different components of the cells and definitely the structure of different organelles. So how scientists have taken out different organelles and how scientists have studied the function of different organelles present in the cell and the composition of cell. For that study, we have to apply different techniques to cell. The first technique is cell fractionation. By the name you can understand, fractionation means putting something into fractions, into parts, a process of separating cell organelle and component based upon size and density. Suppose we have a cell and I want to take out all the material present in the cell and I want to separate that material. So definitely for separating, I will take out different organelles from the cell and putting those organelles separately based upon their size and density. But we will definitely apply a proper technique, a proper method, and obviously with a proper procedure and the use of instrument, I will be able to take out different organelles, or we can say components of the cell. So cell fractionation is a process in which we are going to separate cell components, organelles and cell components based upon the size and density, because there are different organelles present in cell and not all organelles are of the same size and density. So there are two steps. The first step in the cell fractionation is homogenization. Homo means same. What is meant by homogenization? Formation of homogeneous mass of cell. It is just like you put mangoes and milk in a blender and you blend it, and then you get a homogeneous mixture. Like the same phase, the mixture is in same phase. You can't see that solid or the liquid part separately. It mixes well. So in this process, we will put the cells into a homogenizer. Homogenizer is a blender, like you used a blender in your homes, at your homes, for making milkshake or any other thing. That homogenizer is basically a blender. And what will be the function of blender? It will mix all the components of the cell. So what you have to do is you have to grind the cell. It is actually a process of grinding the cells, but in a suitable medium with correct pH, ionic composition, temperature, and the presence of enzyme. Because we know that cells have cell membrane, cells have, like plant cells, they have cell walls. So you cannot just put the cells into a grinder, into a blender or homogenizer, and you just can't break down the cell components. You just can't grind the cell. What you have to do is you have to apply, because these are the cell is a living thing, so you have to apply proper temperature, you have to apply proper ionic composition, and you have to apply enzyme, because enzyme will help you to break down the different components present in the cell wall or the cell membrane of cell. So once the outer boundary of the cell is broken down, definitely the inner matter of the cell will come out, right? The next process is called as centrifugation. You have studied this process, centrifugation, in your books. Centrifugation, which is a process to separate substances on the basis of size and densities under the influence of centrifugal force. In this process, you have to use a machine called as centrifuge, and you have to put that homogeneous mixture you have produced in the previous, we have studied in the previous slide, you have produced in the previous procedure. That homogeneous mixture, you will be there to put that homogeneous mixture in a machine called a centrifuge, but there are tubes named as Eppendorf tube. You will put that homogeneous mixture of cell into Eppendorf tube and then put that tube into a centrifuge machine. Now, centrifuge machine, upon the centrifugal force, will separate the component of cell upon, on the basis of size and density. the more dense the material or the component cell is, it will be in the bottom, and the lesser denser material will be present on the upper side. So you, after centrifugation, you will see there will be a layering in that tube, and that layering will be of the material or the components having more density will be present in the bottom of the tube, and the substances or the components of the cells having less density will be present on the upper layer. So the two different layers will be formed, and there are two type of the centrifugation. Number one, density gradient is the difference on the basis of the density, like the more dense and the less dense material or the components of the cell will be separated. And differential centrifugation, in which you will apply speed to the centrifuge, and the more you will apply speed and the more the less denser substances will be settled down, and the two layers will be formed. The lower layer will be known as palette, and the upper layer will be called as supernatant. So that is how you will see that different layers of these substances, depending upon their density, will be separated out by the process called as centrifugation. The next process of the cell is very easy, and it is differential staining. What is meant by differential? Simple, the word is different. Staining means applying dye or apply a proper stain. So you are actually applying a stain to the cell. A process of applying various color dyes to differentiate between structure of cell. If you have seen the bacteria or any other amoeba or any other cell in your ninth class under microscope, you have observed that there is a different color of nucleus and there is a different color of cytoplasm, because these are microscopic organelles. These are microscopic organism, and organelles are obviously you can't see it without microscope. So in order to differentiate between organelles, in order to differentiate between where is nucleus and which part is cytoplasm, we apply stain to the cell. And when we apply stains, different stain to the cells, we are able to differentiate between the nucleus and the cytoplasm, or depending upon the type of organelle you want to study. So it has two types. Single staining, when you apply only single stain, you want to see the nucleus, so you will apply one stain, so only that part will be stained, and you will be able to determine that stained part is the nucleus, and the rest of the part will not be stained. Double staining, when you want to apply two stain just to differentiate between two different part. Like for example, if you want to apply one stain to nucleus and other stain to cytoplasm, so at the end you will be clearly able to see that this part is cytoplasm and this part is nucleus, because you have applied two different stains, you have applied two different dyes, and that two different dyes will able you to differentiate between two different parts of the cells you want to study. The next is micro dissections. Dissection is basically the dissection is basically a process to separate things, right? To just study for why we are going to separate. By separation we mean we are cutting things and separating it just to study. Micro dissection is the process where dissection is performed under microscopes. So definitely if we want to study, if we want to dissect things, if we want to cut the things to study their internal organelles under microscope, so that thing must be of microscopic level. There are two types of microscopic dissection. Chromosomal microdissection, when you have to cut chromosome or a part of the portion of the chromosome under microscope, and laser microdissection, when you use laser to dissect selected things. For example, if you want to cut a tumorous part of cell, a cell having tumor, and you want to cut the tumor part, Definitely, you have to use microscope, you have to use fine glass needle, and under microscope you will be able to cut that cancerous part from that cell, right? So this process is called as micro dissection.